Good day, Ma'am Russell. I am Novami Pagaling Alsa from BSed Sciences 2. So here is my DNA model. So it was made up of beads, straws, and floral wire. So this portion of my DNA is what we call the sugar phosphate backbone. So there are actually two sugar phosphate backbone to complete the structure of our DNA. This portion, this middle one, in the middle one, this represents the nitrogenous bases of our DNA. It composed of different colors to address or to represent each nitrogenous bases. So the first one is these pink beads. This represents the adenine. These green beads represents the thymine. These yellow beads represents the cytosine, and these blue beads represents the guanine. So this straw in the middle, this red straw, represents the hydrogen band of our DNA. So overall, to attach this DNA, these beads rather, I choose to use floral wire so I could easily stretch them whenever I use this model to replication process and transcription process. So this is my DNA model. The Central Dogma of Life is a process in which the instructions of our DNA will converted into functional products. This central dogma composed of three important processes, namely the replication, transcription, and the translation. The first process is the replication that occurs in our nucleus in the cell, here. So, replication is a process in which it produces two identical DNA which have the same genetic codes here. So, in this process, it needed the presence of DNA polymerase. It is an enzyme that helps this single strand from mother DNA to pair with a newly single-strand DNA. And the other one enzyme that is needed in the replication process is called helicase. Helicase is the one who helps the DNA to unwind, because as we observe that our DNA is in a coil form or structure. So as it pairs with the newly strand DNA, it should have unwind the DNA by the help of our helicase. The next processes after replication is the transcription processes. After the DNA replicate itself into two identical DNA, this DNA will move out from the nucleus and will travel throughout the cytoplasm of the cell. So this each two identical DNA will undergo transcription process in which it needed the presence of RNA polymerase. It is an enzyme that helps this DNA, this newly DNA, produce mRNA, in which this single strand here will serve as the mRNA, in which it composes, instead of thymine, it composes uracil. Yes, uracil. That will bind into adenine. So in these processes called transcription, it will produce mRNA that will later on needed in the translation processes. After transcribing the two identical DNA, those transcription processes will now produce mRNA, which have the uracil nitrogenous bases instead of thymine. So this mRNA will now undergo translation processes in which it will attach in a certain ribosomes, and this process will produce rRNA and peptides. But the most significant products of these translation processes is the polypeptide chains of amino acids. Those are the three types of processes that present in the central dogma, or to complete the what we call central dogma of life.